This tutorial demonstrates how to use the Visual Genomics Analysis Studio VGAS to color plots using heatmap coloring. There are multiple ways a user can apply heatmap colors to their plot. Users can select the plot they wish to apply the heatmap colors to, and then highlight the gene they would like to show expression of in the Genes panel. Then click the button with multiple lines at the bottom right of the panel. This will heatmap cells in the order shown in the sample/cell metadata grid. If the user selects the line button in the heatmap options, the cells will be plotted in default order, being the order that cells were loaded into VGAS. Using the up and down buttons changes the draw order for increasing or decreasing expression, which can more clearly show heatmap coloring. Heat mapping using this option will automatically apply a title to each plot. Another way for a user to heat map a specific gene is by setting the gene as metadata, either in the metadata panel or the sample/cell metadata grid. This method allows more freedom in altering the aesthetics of the heat map. The first thing the user must do is update the coverage depth bar. This shows the distribution of gene expression and is shown in the Color Options panel. The user should then set heatmap color range. This will alter the scale based on the distribution of expression shown in the depth bar. Finally, to set heatmap colors in accordance with the heatmap scale, inclusive of the depth bar and range, choose Set Heatmap Colors by Scale. Setting it by grid will not incorporate the coverage depth or color range. The depth bar and scale can be viewed alongside the plot by ticking Show Heatmap Scale and Depth Bar. To move the scales, select Move, Heatmap Scale, and then use the right click to position and the left click to set. To update plots in real time, tick the Update Plots box in the Color Options panel and ensure that the correct circle, which represents the correct plot and the correct panel that it's plotting from, is selected. The user can then slide the scale to change the heat mapping colors. Their changes will be reflected in the scale on the plot. To change the color of the scale to one of the preset heatmap colorings in the grid, select the desired one and then click Set Scale. Heatmap scale colors can also be selected manually by clicking on any of the 5 colored boxes and selecting a color. A minimum scale value can also be set, with anything equal or below that value appearing in a selected color on the plot. So anything equal or to below 0 would appear as gray. A range can also be set for metadata expression. Once changing a scale, users can also set the scale and save it in the grid.